And the next speaker is Brian Browning from the University of Washington, who will talk about Beagle 5, rapid genotype imputation from large-scale sequencing data.
So this talk is going to present a method for summarizing large reference panels of sequence data, and we'll apply it to the task of genotype imputation. Now, genotype imputation is a term that's used differently by different people, so just to avoid any confusion, I want to make it clear in this talk what we mean. So genotype imputation is the process of turning SNP array data into sequence data. So you have a set of individuals, they've been genotyped, typically for a genome-wide association study on some kind of SNP array. You then combine that with a reference panel of sequenced individuals, and out at the end, you get estimates of the sequence data in those same target individuals. So it's a very inexpensive way of generating sequence data when you have SNP array data. Cost is on the order of a penny a sample. So it compares favorably with sequencing, but the trade-off is for the very lowest alleles, you don't have that accuracy you get from sequence data. So how is imputation performed? So here's a toy example we'll use just to give a flavor of what the methods are. We have 4 reference haplotypes. The variant sites are in bold-faced color. We have a target haplotype. We assume both the reference and the target individuals have been typically statistically phased. So we have phased data. We're going to work on the haplotype level. And you'll see that in this toy example, 3 of the variant sites are genotyped in your target, and the 2 sites that have squares around them, those are the sites we want to impute. The dots represent missing data. Now, in the reference panel, the sites that are not variable, we're not really interested in those sites because if you were to impute it, you just impute them as the reference allele. So I'm going just to remove visual clutter, we'll just replace those with dots, even though they're not really missing data. It just makes it easier to look at. And the way modern algorithms approach this problem of imputing genotypes is we try to represent the target haplotype as a sequence of reference haplotype segments. And you can represent that sequence as just a path like this. And we want that sequence to have the property that every segment of the sequence is, if you were here at the last talk, there was talk about identity by descent segments. We want that segment to be shared identically by descent between the reference haplotype on that segment and the target haplotype. Because if they're identical by descent, which means they've inherited that segment intact without recombination from a common ancestor, then they will have inherited the same alleles, barring the relatively rare events of genotype error or a mutation since the common ancestor. So if they've inherited the same segment identically by descent, then that tells us how to do the imputation. At the first, we just copy the A allele over, and if the second segment is identical by descent, well, then they both will have inherited the G allele. So imputation's trivial. So what's not trivial is determining, what your path through your reference haplotypes is that produces these IBD segments. So the standard way of dealing with that is to consider all possible paths and apply a probabilistic model to those paths. So we take all possible paths, we put a probability distribution on those paths, and then we use essentially the same approach. You want to know what the probability that first missing allele is an A allele. You just consider all the paths that carry an A at that point, because those paths, if they're the correct path, you just copy an A over. You sum up the probabilities of those paths, and that's the probability that the first missing allele is an A. You could do the same thing with a C, and that's how we approach it. Fortunately, there's very fast algorithms for computing the sums of those paths. You don't actually have to iterate over all paths. There's a dynamic programming type algorithm for hidden Markov models, the forward-backward algorithm, that allows you to do this linear in the number of variant sites. And because of the special structure of the recombination, or the switch probabilities, the transition probabilities of the model, you can do it linear in the number of reference haplotypes. So that kind of scaling, if you're in computational work, you're usually pretty happy to have linear scaling. But in this case, just a plain vanilla approach starts getting painful after a few thousand reference haplotypes. So a lot of work, really, the focus of work in the last 10 years that this type of imputation has been around is focused on driving that compute time down. And at the end of the talk, we'll look back and see how successful that's been over the last 10 years. So one of the first ideas, and it's an idea we'll build on in this, it's the idea we'll build on in this talk, is the idea of a custom reference panel. This came from Brian Howey back a couple years after imputation first sort of appeared in the GWAS era. And the idea is to take a small segment of the genome and just focus on that, because in that small segment, very few of your reference haplotypes will share a long IBD segment with the target haplotype. So instead of running your hidden Markov model algorithms, the forward-backward algorithm over all the reference haplotypes, you just sort of ignore all the ones that aren't relevant and focus on that small number of haplotypes that are relevant. This is fairly effective, but it does have some limitations that prevent it from being a complete solution to the problem. So in order to get computational efficiency, you want that window to be as small as possible, because the smaller it is, the fewer reference haplotypes you need to model that region well. But on the other hand, the smaller you get, the more you're getting tunnel vision and you're throwing away more and more information. So this especially becomes obvious when you have really large reference panels, because if you have tens of thousands or hundreds of thousands of individuals in your reference panel, the probability becomes very high that somebody in that reference panel is fairly closely related to your target haplotype. And so they share a long segment identical by descent, but you can't see that anymore. You've got tunnel vision, and you're looking at just that small couple of centimorgan or half a centimorgan region, and you no longer are able to use the information that they actually share a 10 or a 20 centimorgan long IBD segment. So we wanted to develop a method that sort of retains the strengths of this, but is much more flexible, that instead of having small regions, we can use to model with a limited number of haplotypes a whole chromosome. And where we don't have to choose a priori where the window boundaries are. Instead, the data sort of determines the lengths of segments that we use from the reference panel. So this led to the idea of a composite reference haplotype. So here's the standard picture we have. We have 4 reference haplotypes, and what we've done is color-coded the long segments that have shared alleles between the target and the reference haplotypes. And this sharing is at the markers that are genotyped in the target samples. So our composite reference haplotypes are going to be, will be like the target haplotype here. They're also going to be mosaics of reference haplotype segments. So you can see they're also mosaics, just like the target is, but there's a special property they have. We choose them so that if any segment that's shared long between the target and the original reference haplotypes is also shared with our composite reference haplotypes. So you can see that with the green here, you can see that with the purple horizontal stripes here, with the check pattern. Everything's still shared, but because the new composite reference haplotypes are now standing in for multiple of the original reference haplotypes, we can greatly compress the number of reference haplotypes we actually use, right? Because we're enriching them for the parts of the reference haplotype segments that are key to our calculations. So how would you construct these? So I just have 8 or 9 slides here where I'll just go through a simple example that it doesn't capture all the detail of the method, but it gives the key ideas, and it'll give you a flavor for at least one algorithm that's fairly quick for coming up with these, how to construct these composite reference haplotypes. So it's the same situation as before. We have a target haplotype, and I've color-coded the sections where you have long sharing with one of these 4 reference haplotypes. The first thing we do is we just divide it up into small intervals. By default, 1/10 of a centimorgan, which in humans is approximately 100 kb. And then we're going to march along, and as we march along that chromosome, or this region, we're going to construct the composite reference haplotypes as we go. So the first thing is we'll look for matching in each interval. We go to the first interval. We look for a perfect match. We require absolute matching, 100%. At the genotype markers, we find it on the purple, so we just copy the purple over here, and we put an X to make a note what interval the matching occurred in. Okay, so an X goes in interval 1. That'll be important for us in a minute. Interval 2, no match. It's not a perfect match because the target's actually a fusion of 2 different reference haplotypes. Third, we have a match. So we have an open slot. We're going to construct 2 composite reference haplotypes. One of them's open, so we just copy the blue, bring it over, put an X in interval 3 to note that we had a match in interval 3. Interval 4, no match. Interval 5, we have a match. Here's where those X's come into play. We have to choose what haplotype we splice the green haplotype into, right? We're constructing composite haplotypes that represent multiple reference haplotypes. The main question is, which one do we splice it into? So we splice it into the haplotype that has the oldest matching information, the stalest matching. And that would be purple because it last matched in interval 1, whereas blue matched in interval 3. And the reasoning, the intuition here is that we're in interval 5 now. The purple is the least likely to be relevant in interval 5 because it's been the longest time since we've had a match. So we're probably, if there is an IBD segment, it's probably gone. If it wasn't gone, we would have seen a match since then. So we take the haplotype with the stalest matching data, and we splice it in. In the beginning of the splice point, we just put halfway between where the purple last matched and where the green first matched. We've aligned the splice point with an interval boundary here, but you don't have to do that. Go to interval 6, we look for a perfect match. We find it with green. We don't have to do anything here because green's already being copied in interval 6. Instead, we just put an X down to note that, okay, our most recent match for that particular composite reference haplotype is in interval 6. Interval 7, no match. Interval 8, we have a match. So the key question is, which haplotype do we splice into? So if you remember, it was the haplotype with the stalest matching data. That would be blue, right? The blue haplotype has the matching for the longest time in the past at interval 3. So we spliced it in with a splice point roughly halfway between where the blue last matched and where the yellow first matches. So this type of algorithm is linear scaling. It plays very nicely with parallelized code. So you can perform this calculation very quickly. And at the end of the day, you end up with a small number of reference haplotypes that can represent your target sample across a whole chromosome length. And another nice feature is the length of the copied segments adapt to the sharing between the target and the original reference panel. That's very adaptive. You don't have to specify window boundaries or anything in advance. Results. So we're going to apply this to genotype imputation, although it's also in the phasing algorithm in Beagle 5.2, where we found it useful. So with imputation, there's two things we're interested in. With this optimization, we don't want to lose any accuracy, right? Do no harm. And the second is we want big gains in runtime. So first, let's deal with the accuracy, because that's essentially the most important thing. This is a standard type of plot for measuring accuracy. If you haven't seen this before, on the y-axis, it's r², which is a squared correlation between the imputed allele probability, which is a real number between 0 and 1, and the true allele on the haplotype. This is a true allele that we know because we have sequence data and we've masked the true allele. And so we can just unmask it and see whether we got it right. So the true allele is 0 or 1. The imputed probability is a real number between 0 and 1. So r² equals 1, that's higher values in the plot. That's higher accuracy. If you have r² equals 1, it means you're imputing the right allele with perfect confidence, with probability equals 1. R² equals 0 means you got junk, okay? And we have a number of methods here. All the methods except PBWT are pretty much on top of each other, and that's expected because all the methods except PBWT are based on the same probabilistic model called a Lee-Stevens model. So it's not surprising that they're all giving roughly the same answer. PBWT stands for the positional Burrows-Wheeler transform, and it's a little less accurate because it doesn't have the same degree of probabilistic modeling as the other methods, which are hidden Markov model based. The x-axis here is the minor allele count in the reference panel. It's on a log scale. And the reason for that is it blows up in the part of the graph that's most interesting, because pretty soon, once you're up to 80 or 100 copies of an allele in the reference panel, your r² gets very high. So that's not the point of interest, is the low minor allele counts, because imputation accuracy, this always goes down as you start getting fewer and fewer copies in the reference panel. Computation time, this is where we want to see an improvement, and we do. So on the x-axis, we've simulated data, and we went to we exercised a lot of patience to simulate these data sets. This is not trivial to do. Although it's really, with the recent tools, it's at least possible, but a few years ago, it wouldn't have been possible. Reference panel sizes of 10, 100,000, 1 million, and 10 million individuals. These are modeled to match UK-European data. We chose that data set because we have the best demographic models for UK-European data as far as past population growth. So this was the data that we felt we could simulate that was most closely modeling real data. The y-axis is computation time per sample on a log scale. So every tick mark going up that y-axis is an increase in computation of a factor of 10. So going through the methods, first one, impute 4, it has an internal sort of restriction when we ran it that requires the number of reference haplotypes to be able to be stored in a 2-byte integer. So that limits it to 65,000. So we just have one data point, the 10,000. The green is PBWT, and very impressively, it scales to 10,000. The yellow, or I'm sorry, 10 million, and this is, its curve. Yellow is Minimac 4, the most recent version of Minimac 4. It scales better, you can see, because they cross than PBWT. We don't have the 10 million reference haplotypes, although you can sort of extrapolate where they'd end up. We don't have 10 million, not because the software wouldn't hand it. I suspect the software would, but the software, like most packages, has a custom reference file format, and the software tool that prepares the custom reference format, that didn't scale. So we sort of got hung up there. But if that would have scaled, if we could have prepared the reference panel, I think it would have scaled up. Beagle 5 is down here, and you can see its scaling is very well, very good. Like here at the 10 million mark, this is 3 ticks on the y-axis, so you're talking 1,000-fold difference, 3 orders of magnitude difference in computation time. We've done tests on AWS, 10 million reference samples. If you have them, we don't have them yet, but if you have them, one penny to impute a reference or impute a target. It's growing here because there's different section modules of the program scale differently, but even in this last bit, it's still scaling sublinearly. All right, just the last 2 slides, I want to look back at imputation in the 10 years we and others have been developing methods in this area. And I want to benchmark it, I want to compare it against this well-known slide. So this shows the drop in sequencing costs over a 13-year period. The y-axis is in thousands of dollars on a log scale, and it shows a 5-fold, in other words, 100,000 factor decrease in computing costs in 13 years. So I went back and looked at imputation methods and took out some data we had from 2008. And essentially, the time to impute from 60 reference samples in 2008 is roughly equivalent to the time to impute from 10 million reference samples in 2018. That's a 150,000-fold increase in reference panel capacity over 11 years, and it compares favorably, if you can believe it, favorably to the drop in sequencing costs. It's one of the great success stories in computational genetics due to work of several different groups that have just hammered this problem. And the problem's essentially solved now, except for some edge cases. I want to thank my longtime collaborator, Sharon Ying Zhou, who is a fantastic postdoc who did all the analysis, and our funders. Thanks.
These are impressive results, and I'm sure there are some questions. Please go up to the microphone.
Thank you for the nice talk. I'd like to know how much filter you have to do.
How much?
How hard you went to do the filter pre-imputation, and how much cutoff you are going to use for information after post-imputation, please.
Well, it's usually the reference panels have done the sequencing for you, but you can just use standard sequencing filters. Take your data after the standard sequencing filters, phase it, and you got your reference panel. Just standard filters work. The same kind of filters you'd use for a genome-wide association study, or for a study using sequence data. Standard filters will be fine.
Okay, thank you.
Yeah.
Hi, great talk. I was just curious, how do you evaluate the PBWT? I thought it would scale up more favorably than those hidden Markov model-based methods.
Yes, I'm sorry, I didn't catch that word. You thought it was more what?
It should scale up favorably than hidden Markov model-based methods.
That's what you think, but the hidden Markov model optimizations are overtaking it.
Is that because for each target you only have a small limited number of templates to build?
Yeah, there's a number of optimizations coming into play, but the optimizations are giving sublinear scaling for the hidden Markov model algorithms, or at least the most latest hidden Markov model algorithms where PBWT would scale linearly.
Thank you.
Yeah.
Hi, Brian. Thanks for a great talk. You mentioned that you used AWS for some of the 10 million samples, and I guess one of the drawbacks for people who do want to use phasing, who don't have the compute resources, is where do they phase their data? So are you thinking of making the tool sort of a cloud-based phasing tool, or is there an alternative for individuals who want to phase their data who don't have sort of high-performance compute infrastructure available?
All right, so I should point out that those compute times are assuming the data has already been phased. All right,
okay.
so
Yep.
so that's just the imputation time.
Okay, yes.
There are servers out there, like Michigan has a server that
Yep.
will perform imputation and will do some phasing. I don't know how it's tuned, whether it's tuned to give you, the highest degree of accuracy in your phasing or not. I have no idea. It may be. I don't know. I probably won't, just because I don't have the resources to put it in the cloud myself, but I am talking to the people at University of Michigan, and as soon as I have time, we've at least informally agreed that we'll get the phasing tool in place at Michigan.
Excellent, thank you.
So sorry, I have a follow-up question about the PBWT. I wonder how do you run PBWT as imputation methods?
Well, the postdoc ran it, but it was using whatever instructions were on their site.
Well, my understanding is PBWT by default is not an imputation method.
Well, it can be used to perform imputation. I mean, my postdoc was able to do it. I mean, I can put you in touch with my postdoc if you have any questions on it. Yeah.
Time. All right, one last, I guess. Did you look at how the inclusion of rare variants in your input data that you're imputing, how rare variants in your input for imputation affects the quality of the imputation with Beagle 5?
If your phasing is accurate, and generally it can be, the way the models are set up, yes, the rare variants are imputed less accurately, but they don't poison the imputation around them. As long as that phasing scaffold's accurate, each variant is essentially imputed by itself.
Thank you.
Okay, thanks.
In the interest of time, we have to move on. Let's thank Brian again.